Hi everyone, and welcome to this presentation. My name is Mouad, and I'm going to talk about my master's project, which is on the biomechanics of vibrating soft corals. So this project was done at Polytechnique Montreal, and I was with the supervision of Professors Frederick, Gosselin, and Stéphane Etienne, and was funded by the INSERC and the CBS. So the starting point of this project was actually an observation made in the West Indies, where a soft coral was swaying back and forth with the effect of the surface wave, but at the same time was exhibiting some small and rapid vibrations at the level of branches that you can see here in this video. And that's an intriguing motion. It has been reported just recently, and we have no clue of what it is or what are the underlying dynamics. And the goal of our project is to understand more and get insight into this motion. From a mechanical lens, you can see this coral as a flexible structure vibrating with multiple frequencies, and therefore we can study this system with mechanical concepts and more specifically using fluid structure interaction concepts. So I would like to start first with the introduction of the species we're working on. So that's the bipinnate sea plume, and it belongs to the family of soft coral, which is a family very well known for their diversity of colors and morphologies and so on. And they also belong to a larger family of Cnidaria, which encompasses sea anemones and jellyfish, for the reason that if you zoom into a branch, you're going to find some small organisms called polyps, and these have tentacles and the mouth and the digestive system, etc. And basically they are sharing the same anatomy as jellyfish. These corals are animals, they are not plants, even though we're participating in a plant biomechanics session, but don't tell this to physicists, they will kick me out. But anyway, so these corals need to eat, and beside photosynthesis, they rely on food particle. So when a food particle, when a plankton or a larva, for example, approaches the polyp, well, the tentacle stretches and catches the food and brings it down to their stomach. So if you're seeing this vibrating coral in a scuba diving or a documentary or whatever, you might probably ask yourselves, well, why is this soft coral vibrating like that? And of course, that's a very big question, but we're going to treat only two aspects of it. First, we looked at the origin of these vibrations, and then we studied the impact of these vibrations on the food capture and the biology of soft corals more generally. So regarding the origin of these vibrations, we proceeded like an experiment. We extracted or estimated, more specifically, the most important parameters using ImageA, and we computed the most relevant numbers, which are the Reynolds number, which was around 200, and reduced velocity, which is around 5. And these two values are actually clues telling us that the vibrations we are seeing here are most likely coming from vortices downstream the coral. And we are talking about the vortex-induced vibrations instability. VIV are very common in FSI. They appear when you have a vortex street forming in the wake of your structure. And what's special with these vibrations is that when you see the amplitude versus the flow speed, you're going to see that you have a critical region in the middle where the amplitude is very high. And if you look at the frequency of your solid, that's an increasing function of the flow speed, but at this critical region, that's a constant and equal to the frequency of the vortex. And we are usually saying that the frequency of the solid is locked into the frequency of the vortex, and this region is subsequently called the lock-in region. So to verify our hypothesis, we simulated the motion of an elastic rod subjected to VIV, and for that we wrote a Python code which is based on the finite element platform Phoenix, and we implemented the wake oscillator model proposed by Facchinetti and colleagues, which is a very well-known model in FSI, and the results are the following. So basically you're seeing that both dynamics are quite comparable in both worlds. So you have similar amplitudes, and durability is also quite similar. So, and this result is actually a strong argument telling us that what we are seeing here in the ocean are most likely coming from vortices. All right, so now let's move on to the second part of this presentation. And here we want to study the effect of these vibrations on the particle capture. Here I'm showing you an image of some living corals we've bought in our lab, and as you can see, there are a lot of biological complexities. There's a special branching pattern and textured skin, etc. And we as engineers, we love to simplify things, sometimes drastically. So we took a single branch and considered it as a circular cylinder, which is mounted in two springs so that it can move in both directions. And the particles are considered as spheres and also non-motile, so they can't change actively their trajectory by swimming, for example. So our first thing we did is to simulate or compute the flow around this cylinder, and for that we use an in-house finite element solver called CADIF, and it basically solves the Navier-Stokes equations using direct numerical simulations. And as you can see, we have clearly the formation of vortices and vibrations as well. The second thing we did is to take this flow solution and insert it into a Python code we wrote again. And this code actually takes each particle and computes the total hydrodynamic load exerted on it, which is composed of the drag, the pressure load, and the added mass force, and integrates this Newton's second law, basically. And also here to mention that the capture criterion is simply a solid contact between the cylinder and the particles. So to evaluate this process, we need to calculate the capture rate, which is by definition the number of captured particles per unit time or per second or whatever. But it's also equal to the flux of particle entering through this orifice that we call the capture window. And that's the region where all the particles would be ultimately captured. That's a very practical definition for our computations. And of course, this capture rate will depend on a lot of parameters, but we can reduce them into just 5 using dimensional analysis. And also, we will simplify further our problem by considering only 3 parameters, which are the diameter ratio, the Reynolds number, and reduced velocity. And also, we only take the time average of the capture rate, eta, in this dimensionless capture rate. So here I would like to start with a simple case of the fixed cylinder, because we are turning off this UR, which is the variable representing vibrations. And here I'm showing you the evolution of eta. And what we are learning here is that the capture rate increases with the size of the particle, as well as with the Reynolds number of the flow. But there's a second interesting thing here, which is this power law we've got here. We have a scaling. And to see the difference between these two scalings we've got here, I'm showing you here the trajectories of particles. So for small particles, you can see that the trajectory is quite straight at the beginning, and then abruptly turns near the stagnation point here, and then gently continues its path. Whereas for the larger particle, it deviates earlier and then impacts more strongly the cylinder. So in this project, actually, we're curious into deriving theoretically this power law. And for that, we thought about an idea. We said, well, the particle is so tiny, so it's actually seeing the cylinder as a flat plate, right? So based on this idea, we took our cylinder and we transformed it into a flat plate using the Joukowsky conformal transformation. And then we applied the Blasius solution over the boundary layer. And with some algebra, we ended up with this scaling. All right, so now for the vibrating cylinder, we calculated the capture rate eta for various URs that you can take as a sort of dimensionless period of oscillations, if you like. And so we've got these results, and you see that we have a bell-shaped curve with a peak around 5, and also tails plateauing towards the case of the fixed cylinder. And this result actually is telling us that if you are in this optimal region near 5, well, the vibrating cylinder is doing a better job than the fixed cylinder in terms of capturing particles. And we did that for other Reynolds numbers, and we've got the same pattern. We always have this bell-shaped curve. And if you recall the curve of the amplitude versus the flow speed during VIV I've shown you earlier, that's also a bell-shaped curve. So it seems that there's a sort of correspondence between the capture rate and the motion of the cylinder, and indeed that's what we found. We've got some correlations between the kinematics of the cylinder and the capture rate. All right, and here I'm showing you the gain in capture in percentage, and we're interested in the value of the maximum. And here's maybe the most important conclusion in this project, which is simply vibrations are beneficial and good for capture. And that's an important conclusion, partially because it flips this old and dark view, saying that vibrations are bad, they are destroying our buildings, our infrastructure, and therefore we need to prevent them. Here we are showing that vibrations are not completely evil, and sometimes they can be beneficial, and therefore we need to trigger them instead of removing them. Okay. So here just quickly a few words on the biological implication of this result. So I've said that if you want to score a good capture, you need to have a UR near 5 in this optimal zone. So the UR for a rigid cylinder in our model has this formula, but if you think of a real coral branch, we can show that it depends on the aspect ratio of that branch and also the Young's modulus of the coral skeleton, if you like. And this formula is actually shedding light in an important phenomenon in biology called plasticity, which means that if you have a single species and you put it in two regions in the ocean, well, you're going to end up with two different shapes and morphologies. So for example, if you have a coral and you put it in a region with turbulence, a very high flow speed, well, you might end up with very high URs, and that's not very beneficial for capture. But if the coral decides to reduce its aspect ratio or increase its Young's modulus by maybe 18 more calcium carbonate components, for example, well, the UR might be shifted back and reduced toward this optimal zone. And it seems like the coral is feeling or sensing its surrounding environment. Okay. All right. So in a nutshell, there are 2 points that you need to remember from this presentation. Soft corals vibrations are first induced by vortices, and then they are good for particle capture. Yeah, that's right. So thank you for your time.